At the end of the Ordovician was the second biggest mass extinction since the rise of animals. Amazingly, in South China we actually have a spectacular window into what life was doing in the immediate aftermath of this event. There's a lot of work left to do to fully describe and understand this, but here's an introduction to the Anji Biota and how it is revolutionising our understanding of both sponge evolution and the history of deep ocean communities. There have been 5 great mass extinctions since the start of the Cambrian. The biggest of the lot is of course the Permo-Triassic catastrophe, also known as When Life Nearly Died. Thanks Mike for the catchy title. But there are some lesser ones, like for example at the end of the Cretaceous, which bumped off quite a lot of the dinosaurs. But the other really big one was actually at the end of the Ordovician. This was the result of, or at least correlated with, a very short, sharp ice age, a series of glaciations that were quite spectacularly deep, and then a very sudden warming again. So you have gradual cooling through a lot of the Ordovician, then a sudden actual glaciation, sea levels dropped the works, and that lasted for about 1 million years before the ice retreated again and it went back to relatively more stable conditions. It took a while for life to recover though, and the extinction itself wasn't at the very end of the Ordovician. There was a few 100,000 years, more or less, before the beginning of the Silurian, as we recognise it, and then you have the start of the recovery period. But those few 100,000 years at the very end of the Henantian, which is the last interval in the Ordovician, are basically dominated by black mudstones. There were a lot of problems with the ecosystem. There was ocean anoxia. It was not a very productive time. Most things didn't start recovering at all then. Graptolites showed the beginnings of a recovery. They started to rediversify, but they were living up in the plankton. On the seafloor, there were all sorts of problems with anoxia, circulation patterns going haywire, and as a result, there's not that much in the latest part of the Ordovician. It is, of course, an extremely interesting time for graptolite specialists, though. And when we lived in China, this was back in 2013, and just before we left there to come and try to make sense of our lives and get a proper job and things like that, we had some field work in the beautiful Zhejiang Province, which is forested mountains covered in bamboo, lakes, deep valleys, beautiful place. We'd actually gone there mainly to find more specimens of what we later described as heteractinigma. This is a bizarre sponge that I've mentioned in the hexactin problem video, and I'm sure I'll do more about again in future. But went back to that section to collect more, found more sponges, but no more of that. But anyway, at the end of the trip, our colleagues, particularly Zhang Yuandong, decided that they wanted to do some of their own field work since we're in Zhejiang anyway, and went to a place called Fusha Reservoir, which is one of these absolutely stunning little idyllic places, a reservoir deep in the bamboo forested mountains. And there was an interval of black mudstone on the shore of the reservoir, which was from this interval after the mass extinction and before the Silurian recovery. So it's actually quite a thick sequence. There's about 10 metres of mudstone there exposed on the bank. And they wanted to log very carefully the graptolites in each layer as part of an ongoing project, so just something they needed to do, and work out what the pattern was. Which species were evolving, which were going extinct, what the sequence was. And that's basically the nuts and bolts of what a lot of paleontology is about. You go out and you do careful work, finding things, describing them, and putting together a historical sequence. So we were at a bit of a loose end. It was the end of 2 years in China, at the end of a field trip, and we were a little bit tired and, just wanted to sit and admire this gorgeous, stunning scenery. So I sat myself down on the side of the reservoir, just sat down on some of these rocks here and just sort of admired the birds singing in the trees, and it was lovely. But, as a paleontologist, you have slightly twitchy hands, so there was a lump of rock next to me that looked a little bit loose, so I just sort of pulled at it a bit and didn't even bother getting my hammer out, just sort of whacked it on the ground. broke open, and there was this finger-shaped thing on the rock, with a hole at the end. Something like this, actually. There's a couple of them there, but yeah, finger-shaped things. It looked remarkably like a sponge. So I sort of picked up the next bit next to it and broke that as well, and another sponge, and another sponge. Sponge, 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 sponges everywhere!
This is a basal Silurian graptolite sequence, and there's this layer which seems to be absolutely packed full of Gazillion sponges, which is rather fabulous. To give you an idea, is part of one long tall conical thing, and then there's other bits like these, a little one on top of another long one, and a conical one there next to it. So
do anyone else would just look like blobs? How do you know they're sponges?
They've got spicules. Eagle! Ah, an eagle!
Ah, an eagle!
Oh, thank you! That is good to see.
This is not what you expect in the Hernantian. There was something like 3 species of sponge described from the entire Hernantian worldwide. And they were all sort of limestone, shallow water things. So in about half an hour we collected probably 10 species of sponges just randomly from this one little bit of the outcrop. This was slightly surprising. Unfortunately, as I've just mentioned, we were just about to leave China and go and work out what to do with the rest of our lives. So we put these sponges in some trays and left them in Nanjing. It was a few years before we went back there to do any more work, and then we started doing a series of short-term fellowships, which we still go out for every now and then, and normally just a couple of months, 2 to 3 months at a time. And since then, since about 2015, we've been going back quite a few years and have been working steadily on the Anji Biota. Because in that time period between us leaving and coming back again, it was a chance for Zhang Yuandong, our colleagues, and to work with the local Geological Survey, particularly a lovely chap called Wang Longwu, who has been working in that area and was a very keen paleontologist, or amateur paleontologist, and he was out looking for more of the exposure of this interval of black mudstone and possibly other sponges. So when we got back there, not only did we have this section on the side of the reservoir, which was now pretty much entirely flooded, such is life, but not only that, but we now had probably 6, I think it was at the time, other sections which expose this black mudstone interval. And we started to piece together a quite decent sort of story about what this represented, where it fits in the sequence, and the sponges, it turns out, were all over the place. I should probably say that I'm actually drinking a cup of the Anji Biota, which is the Anji white tea, that is grown on the outcrops, and you can practically taste the sponges. What could be better? We actually buy this directly from the farmers, and we quite often get lunch from them as well, just sort of turn up and say, can you fit 8 people into lunch? And they say, oh, maybe. And yeah, anyway, you get to know the locals quite well when you're going regularly on fieldwork to these places. And it's a very sort of intimate rural sort of community. Lots of small villages, mainly based around bamboo and the tea growing. So there's lots of, well, I say there's lots, there are some roads winding through these forests. And it's usually on the road cut sections where we find decent levels of rock exposed. And the Geological Survey in particular, as well as our colleagues, have been doing a lot of work piecing together the sequence, looking for graptolites to work out the ages. And what we now have is approximately 360 metres of sandstone turbidites. These are thick beds of sandstone that slump down undersea slopes into deeper basins where they're deposited as discrete layers, mudstone between them, then another turbidite. And most of the Hernantian, most of the Late Ordovician in fact, but the Hernantian especially, is represented by these thick turbidite layers. you don't get much in them. There are occasionally some graptolites in the mudstone partings between the layers, but mostly this is effectively barren. So you can trace the age quite neatly from the little graptolites that you do find. So we know that we're well into the Hernantian at this point, and then there are two biozones within the Hernantian, so you can recognise the Extraordinarius zone, and then the last part is the Perscoptus zone. When you get a certain distance through, about 300 metres through these sandstones, suddenly there are 10 metres, more or less, of black mudstone. It weathers quite pale, that sort of colour, and it is also covered with graptolites in a lot of places. Not every layer, but a lot of it is just layer upon layer of thousands of graptolites. And I'm sure Lucy will tell you a bit about them in a bit. But this black mudstone interval has some turbidites at the bottom, which fade out. Then it's dominated by mudstone for a short time, and then turbidites come in again, and it's replaced more and more by these thick sandstone layers. So it's a short interval. And this is generally interpreted as representing a point of relatively deep sea level, where the sea level has risen again after the glaciation. So the sandstones, the turbidites, represent the low stand of the sea level. That was the Ice Age. That was when you get not much going on in terms of biodiversity. You have a fauna that was specialised for cold conditions, and then after that you have a sudden reversion to warm conditions, and you get the sea deepening, the sandstone is replaced by black mudstone, and you get circulation problems with anoxia. So black mudstone, lots of burial of carbon, lots of plankton blooms going on the surface, sink down to the bottom, and not much is decaying it because there wasn't much oxygen on the seafloor. And then gradually the sea level fluctuates a bit more, and you end up with it falling a little bit again, more sandstone coming in, and it gradually builds back into a normal pattern in the Silurian. So this mudstone interval appears to be the time of the local deepest water, the local highest temperature in the aftermath of the extinction. So the graptolites tell us that it was still in the Ordovician. We haven't reached the Silurian yet. There's no recovery going on. But at the base of the mudstone, there is one interesting little deposit. It varies between sections. It's up to 1 metre thick. Sometimes it's not visible at all. But this is a strange green clay layer that contains a shelly fauna, little brachiopods, bits of trilobite, Macaridian sclerites, for those who know what those are. If you don't, I'll do a video on them at some point. And a range of little shelly things, but no graptolites and no sponges. And that is a transported slumped layer from shallow water, which represents a deeper water equivalent of the Hernantia fauna. This is a famous cold-adapted community that occurs worldwide effectively with variations. And this is the Aegiramonella fauna. It's a deeper water lateral equivalent of the Hernantia fauna. And it occurs just in this slumped green mudstone that we get at the base. And then from there it goes abruptly into the black mudstone, where we have a very different sort of community. I should say the sponges do not dominate all the way through. But in each section, and we went to all the ones we could possibly find, there are intervals where you have truly abundant sponges. It's the sort of place where you take a lump out the outcrop like this and you split it and there's a dozen sponges on there, split it again, there's another 20. It's just endless. And a normal sort of bedding plane assemblage is something like that, where you can see it's just sponges all over the place. And quite often it is one species dominating, so this thing is what we call the finger sponges, which actually named as Shoja which means finger sponge. But then when you start looking and collecting intensively, you find that there is a lot more to the diversity than these one or two abundant species. In fact, it rapidly starts getting ridiculous. I should say now that there is not a great range in the diversity of different groups present. So, you know there are sponges, you know there are graptolites. Aside from that, we have nautiloids, not very many of them. These are the ancestors of ammonites and things like that. They have a conical shell. We can't work out the species because they're not well enough preserved. I think when we published the first paper, we had something like 5 had been found in all of our fieldwork. But now there are quite a few more, and there's one section when they're relatively common. But nautiloids are, in most of these black mudstones, really quite rare. We also have one eurypterid, a sea scorpion. It's the first Ordovician one from China. It's a partial specimen going off the edge of the slab, and we found that on our first set of fieldwork when we went back in 2015 or so, and we have never seen another one. We do also have some more enigmatic fossils. So we have rare examples of bivalve arthropods, carapaces and tail spines and things like that. We have some very interesting ring-like structures which seem to be a ring of plates. And we have been thinking that these are radiodont, but basically everyone is saying that we need to get better material, ideally an appendage, before we can prove it, which is probably fair enough, but that's the only thing we can think that they can be. There are also some other things that we're turning up now, nothing dramatically exciting, possibly, but potentially very interesting. But we're keeping those under wraps for the moment until we actually can do a bit more work on them and find out what we've really got. But that's it. There are no other mollusks, no bivalves, no gastropods. The only gastropods are in green clay at the base. There are no trilobites. There are no brachiopods. None of the normal fossils that you might hope to find in the late Ordovician have turned up. But in terms of total diversity, it's probably not far off the diversity known from the Burgess Shale. That sounds like a ridiculous thing to say, because we only have something like 20-odd species of graptolite, and the Burgess Shale has been collected for over a century, and there's well over 200 species there. The point is, though, that we think there are probably well over 200 species of sponges in the Anji Biota. I don't know an exact number yet, because it's still ongoing. Every time we go back, I spend the first month simply writing up. So I'm working up towards a monograph, basically a book. I've written up about 100 species now, which is taking up something like 300 manuscript pages, not including figures. It's quite a lot of work so far, and I think I'm about halfway through what we've collected. So every time we go, I write up another 20, 30 species, something like that. Almost all of them are new, by the way. And then we go on field work for a week and we collect another 40 odd new ones. So it just feels like every time we go, I'm getting further and further behind. And at some point, I'm just going to have to stop, publish what we've got as a sort of monographic or semi-monographic work, and just accept that there'll be a second instalment of a monograph sometime in the future. Because this diversity is getting ludicrous. It is far more diverse than equivalent faunas in the modern deep ocean. How deep do we think this was? That's actually a really difficult question, because absolute water depth is one of the hardest things to try to work out when you're a paleontologist in general. If you've got shallow water indicators that tell you you're in the photic zone because you've got photosynthetic things, no problem. If you've got sedimentology indicators that show the action of waves, no problem. You can pin those down to the shallow water end quite easily. But beyond the photic zone and beyond the shallow water conditions, you're looking at sort of general interpretations of the tectonic setting, what the likely seafloor depth is in that sort of setting, and so on. And what we can say for this is that it is much deeper than we normally expect to see. Most of the fossil record is deposited on the shelves. These are bits of continental crust that surround the land masses, and those are the bits that are steadily built up, turned into mountain ranges, and get preserved for posterity. The deep ocean, the abyssal plain, is on oceanic crust, and this is normally subducted. It sinks underneath another continent, gets destroyed back into the mantle, or it gets crunched into continental collisions and turned into mountain ranges and metamorphosed. So we do see bits of the slope, the transition between them, but even that is relatively uncommon. Normally what we're seeing is either the shelf itself or relatively shallow basins where you have the crust has been sagging a little bit due to tectonic forces acting on it, and it gets pulled apart and the crust sags and then gets filled up with sediment. We do have some of those. But in this case, we have quite a lot of direct evidence that it is below the edge of the shelf by some margin. So, for example, we have the deposition, the preservation of these sponges is in layers of mudstone that were deposited as collapsing nepheloid layers. We know this because there are little pale blebs of mud throughout most of these beds, and the distribution of the orientation of the sponges, they're mostly not oriented, so there isn't a consistent current that's washing them down slope. They're in all sorts of layers. They can be locally oriented, and then a millimetre above them they're at 90 degrees to them. But it's really the fact that you have lots of these little blebs of pale organic pore mud material. And this is what we call flocks. It's basically where you have bits of clay particles in the water that combine through electrostatic attraction and then sink. And you get massive amounts of this in nepheloid layers. This is where you have sediment coming off the shelf, hits normally around the shelf edge, and then that sediment forms a layer floating in the water, which creates a density boundary. And it floats off along this density boundary. So the deeper water is colder, more saline, denser, and then you have this layer of a sort of cloudy layer, a nepheloid layer, floating off at that sort of depth. And eventually these electrostatic charges make the particles clump together and sink. So any disturbance and the whole thing just goes flump onto the deep ocean floor. And that appears to be, at least based on all the lines of evidence we've got, that appears to be how they're being preserved. So that's one thing. Another line of evidence is some of the nautiloids are very, very weirdly preserved indeed. They're broken. We find them on bedding planes, jointed at several sort of angles. And to start with, we're seeing these and thinking, is this some sort of elongate arthropod? Every now and then we still have doubts, but no, we're pretty certain these are nautiloids that have been shattered by pressure. So as nautiloids sink through the water column, there are chambers inside the shell which contain gas that the nautiloid itself used to orient itself in terms of buoyancy. So that's how it goes up and down through the water. It can secrete gases into these chambers. But they're mostly sealed. So as it sinks, the outside pressure increases and the chamber walls gradually collapse as the pressure increases. In extreme cases, though, they don't see many examples of this in the fossil record at all. They can completely implode. And this apparently tends to happen at significant depths. So we're talking 500, 700 meters, something like that sort of depth. You start to get complete implosion happening. And that seems to be what is happening here. The other line of evidence is based on the sponges themselves, because we have a diverse array of hexactinellids. For those who watch the hexactin video, I don't mean things that have hexactins, I mean real hexactinellids. In fact, I mean living families of hexactinellids. Things that today are placed in the Rosselidae, the Euplectellidae and others, living families of deep sea sponges. Exclusively deep sea. These things live, with a couple of minute exceptions like under Antarctic ice, entirely in the deep oceans, the abyssal plain and the continental slope. So normally these things are 1,000 meters down. And what's most remarkable about these sponges is that there is no other fossil record of these groups at all until you get to the Jurassic and Cretaceous especially. That's about 100 million years ago you start seeing them again. And here we are at 444 million years ago. the entire intervening time, there is no sign whatsoever of these groups of sponges. That's quite extraordinary, because today they live in the very deep ocean, and the fact that we do not see them through all of the rest of their history suggests that they also lived in this habitat through most of their past. So the Cretaceous was a very odd time. We'll come on to that some other time. So that's another line of evidence that we're looking at a community, an entire ecosystem that is normally missing from the fossil record entirely, because they live too deep down. There is another potential explanation, though, for having lots of sponges in a place where you don't normally see them at this time, because we're right after a massive great mass extinction. So that is going to affect things a little bit. And is it possible that you have an entire community of sponges that found an empty ecosystem above them after everything had died, and just crept up the shelf, in sort of ecological terms, not individually, and started to colonize areas where they would normally have been excluded because they were not just too shallow, but there were other things living there? Is it possible that this is an aberration, that it's just because of the mass extinction that they have taken over shallower conditions and we're not seeing anywhere that deep at all? Well, that is contradicted by the nautiloid implosions and the nepheloid layers, but still, we have to look at that. And it turns out that in South China, it's not just the Anji Biota. At the same time as we were writing this up, Li Xia and colleagues were working on what they call the Beigong fauna. This is from about 100 kilometres away at pretty much exactly the same time, and was also dominated by sponges. Interesting. This actually led to a bit more work, because we also had reports of another site which we went to look at Gangangshan in Jiangsu Province. So this is another 200 kilometres further north. And there we also found sponges in one of these boundary intervals. So we ended up working with Wang Wanhui and colleagues as well, and trying to map out where you get these sponge-dominated, rich sponge communities around China. South China at least, South China being a separate continental block to North China. And it turns out that at this time, at the end of the Ordovician, they were absolutely everywhere. Every time we get a mudstone sequence that crosses the boundary, we either get complete sponges, or we have disarticulated spicules forming dense layers which show that there were abundant sponges there. And we managed to extend this across 2,000 kilometres. Even more recently, Zhongruo Wen and colleagues have just published the Huangshi fauna, which is from the middle of South China and seems to be even more like Anji than some of the others. There looks like there's some soft tissue preservation going on, and there are also arthropods and nautiloids. So we get to see what else is going to come from there and how diverse it is, but that's yet another site. So it turns out that when you start looking, they're all over the place. We even went, slightly embarrassingly, to a place called Wangjiawan, which is the stratotype for the Henantian. This is the section that defines the base of the Henantian. And in China, these things are celebrated, and they are intensively studied. They have been analysed millimetre by millimetre to look at the changes in graptolite faunas as you go through the Henantian and into the Silurian. And there had never been any mention of sponges there. And the section is protected, so we couldn't actually hammer the outcrops, but it took us about 10 minutes to find complete sponges in the loose blocks at the bottom of it, which we can then tie in by the sedimentology to the same interval, latest Ordovician into the Silurian. So it does appear that sponges basically took over at least the offshore shelf communities and into the basin around the end of the Ordovician and the early part of the Silurian, before they gradually were displaced again by other communities starting to recover. That was quite remarkable, and this actually led to our first couple of papers, 2017, 2018, when we were describing this as a general pattern. Because when you start looking into the literature, most of the other mass extinctions also show a proliferation of sponges immediately afterwards. It appears to be a general pattern that when you have mass death in the oceans, some things don't mind at all. So you imagine you're a sponge sitting there filtering organic particles out of the seawater. You're quite tolerant of things like low oxygen conditions because your metabolism is fairly low. And what you end up with is a large number of sponges. They absolutely love it. It seems like mass extinctions are something that just generate lots of decaying organic material in the water, and that all gets soaked up like a sponge or something. Yeah, basically, sponges love it. So we're seeing effectively a disaster taxon. It's like on land we have famously the fern spikes after mass extinction, because they can tolerate these conditions of really cloudy, disturbed weather, and they thrive when other plants disappear for a while. And in the ocean, sponges do the same job. So that was really interesting. And this is part of ongoing studies. Other people are recognising this more and more around other extinction events now as well. But in terms of, does that mean that the Anji Biota isn't deep water? Still no, it doesn't, because what we get are different sponges. The ones that we find all over the rest of South China are the ones that we get at the top and bottom of the Anji sequence, where we get a diversity of more primitive sponges. So earlier branching groups, things like ascosponges, reticulosans that may or may not be ancestral to hexactinellids, or at least directly ancestral. But the really diverse fauna of the Anji Biota, these extraordinary true hexactinellids, the rossellids, the euplectellids, things like that, are still restricted only to Anji. Of course, the sponges are the most important group at Anji, but the graptolites are actually the most abundant. There are just so many bedding planes absolutely covered with billions of the things that it would probably be quite bad of me to ignore them. So while I've been writing up the sponges, Lucy's been going out to write up the graptolites, and she's nearly there, so she can probably say something sensible about them. Maybe. Hang on a minute, let's find out. Well, I dragged her out of her office, just for you. So, Lucy, the sponges are completely different in the Anji Biota to everywhere else.
Oh yes.
Is that true of the graptolites?
Generally not. I mean, the nice thing about graptolites is because they lived in the water column, floating or swimming, we'll get into that another time perhaps, you tend to get consistent graptolite faunas all around the world. So the species of graptolite I was getting in Anji were generally, in almost all cases, ones that were known from elsewhere, either in China or other places like the US, the UK, Australia, really anywhere where you've got rocks of the right age. And of course the consistency of faunas is why you can use graptolites for dating rocks. This is why sponges are totally useless for dating rocks. I don't know why he's looking so proud there. It's not a thing to be proud of.
Oh, it is.
So, which is why he needs me. So yeah, so it's about, I don't know, 15 odd species in the Anji assemblage, and a lot of them, almost all of them, we're getting elsewhere. What's interesting is that we don't get at Anji that there are some quite distinctive species known in the shallow water at this time, things like Researchgate, which has got lots of spines up it, which even quite a small fragment of that would be recognisable, and we haven't had a single one there. So it looks like maybe the spiny things are in shallow water, and that fits with what we've seen elsewhere in rocks of different ages. So that's something we might want to do more about at some point. And there are more graptolites in Anji than there are sponges. It is in fact a graptolite-dominated fossil assemblage.
I've already said that. No getting away from it. But we don't need to celebrate it.
You don't, I do.
Right.
So we've got thousands and thousands of graptolites, maybe tens of thousands I haven't dared to count, and we don't even bother collecting all of them, that's how abundant they are.
So how many new species have we got?
Two.
Two?
Both of which have been described in the literature.
Mm-hmm.
There's a new species of Vitagraptus that actually tells us a bit more about how that particular group of graptolites evolved, so that's quite nice. And then there's an entirely new genus, which is basically one that's a V shape that. Have you shown us pictures of graptolites?
Not yet.
Ah, he'll show you some pictures of graptolites.
Here's one I prepared earlier.
Here's another one. Just to make things difficult for you.
Yeah, thanks.
So almost all the graptolites at this age, they're two branches but back to back, so you basically got a single graptolite unit with tiki on both sides. The thing that are called Anji Wangi starts off like that, and then the two branches split and become separate, so you get a nice sort of, well, Y shape really. And we got this one specimen discovered by one of our colleagues from the Zhejiang Geological Survey, Long Wang, and got very excited by this because, you know, something different. And so we went back to the site the next day to try and find more, because that's what you want generally with fossils, you want more than one. And so there were about 6 or 7 of us at this one site all day. Not a single other graptolite. Well, lots of other graptolites, but not a single other thing we eventually called Anji. And so I published a paper a few years ago, and then one turned up last year for a different site.
So let me just get this straight. Of all the graptolite workers working all over the world, this species had never been seen before by anyone, or at least not reported.
That's right.
And it's quite distinctive.
Oh, yes.
And in 10 years of quite intensive fieldwork, and we have collected an awful lot of rock over those years, we have 2 specimens of it.
Yes, that's right.
How exactly does that work ecologically? How rare was this in life? Because it should be preservable in this environment if it was anything like common.
It would be very preservable. It would be as preservable as any other graptolite. It's not that. Either they were present in incredibly low abundance in the life assemblage, or they were living in a different environment and you only get a single occasional one swept in. So maybe they were even further out to sea.
In even deeper water. But we're in pretty much the deepest part of the basin in Anji. That's why it's got this fauna.
Well, it doesn't have to mean living in deeper water. It could have been living further out to sea.
But that gets us
But
onto
in shallower water.
But that gets us onto the Yangtze platform, Gangangshan and so on is further. That's further north. The coastline is to the south. Further out to sea is.
Well, further out into the main ocean.
Oh,
where the sediments are not
right.
preserved.
Okay.
Yes.
So you'd be looking at.
Yeah, you just get the occasional one swept in by a storm or something.
In the direction of Australia, I think it was at the time.
Oh, probably. Yes.
Which has been well studied for graptolites. But it could be.
There's a surprisingly little rock of that age in Australia, I think, that's got graptolites. It's not that many assemblages described.
Okay. Okay, so you could have something which is planktonic.
Mm-hmm.
It's floating around
Or swimming.
at the mercy of the currents.
Mm-hmm.
And yet it doesn't normally drift into even the continental basins, let alone the shelves.
Yeah.
Because if it did, it would have been found.
Yes.
It's actually really interesting how that can happen, isn't it?
Yeah, I mean, there is a lot we don't understand about rarity in the fossil record.
Yeah.
And probably, well, you think of the vast unlikelihood of any single thing first, becoming fossilised at all, and then being found by a paleontologist who actually does something with it. And those 2 steps can, a lot of stuff can get lost in those last 2 steps.
Yeah.
The chances of getting any individual as a fossil are very low indeed.
Yeah, I'm particularly astonished for graptolites at how they can be so rare and yet have survived, because you have the problem of how do they reproduce? If you've got such low population density of things which weren't particularly active swimmers, even if they could swim a little bit, they can't really find each other. So presumably they were reproducing via spawning of gametes into the water or something like that, but...
Presumably, or they were obligatory self-fertile, they only ever mated with the same colony, which seems a little unlikely too.
It does, because then how could they actually evolve so dramatically? Without much mixing? But yeah, that's another question,
That's
I guess.
a very good question, but let's not.
So the question is, if you've got something which is so rare in these ecosystems, how do they ever find each other to reproduce?
It's a problem, isn't it? Which is why I suspect it was quite abundant, but somewhere where we haven't got any rock record.
Yeah, but that leads to other problems of how did they stay out of these basins, mostly. Yeah, I'll do another video at some point on what rarity actually means in the fossil record, because when you've got the entomology perspective as well, it's an interesting question.
Mhm.
Okay, so I've been spending the last 10 years of visits writing up, a couple of hundred species of sponges.
Yes, you have, haven't you?
So why did it take you a bit longer?
Well, it's because, almost all the graptolites have been described before. That if I find a graptolite specimen, or a group of graptolite specimens that I think are the same species, I have to look at everything that's ever been published to work out what it is. To work out if it's... I can stop, when I find something that's the same. And then, I can go through the existing literature because I know the name then. But if I think I've got something new, I have to go through everything else. Hundreds of species, checking that it isn't an already known one. And I thought I had a third new species. I got really excited. It was a lovely little thin thing and nice material and got good illustrations and wrote up description and was writing a very long remarks section in my systematic section about why it isn't all these other very thin little graptolites that look similar. And I thought, I'll just check this last one. It's a bit of a different age, so it's probably not that. And it was the same thing. And at least I noticed before it got to actually submitting the paper, because that's a bit embarrassing when it comes to the reviewers pointing it out, which they would have done.
Giving the same species a different name is generally frowned upon.
It happens all the time, but we really should try and avoid it.
But at least you've got all this vast literature out there, you've got a wonderful library, you can just go to the paper and tell immediately if it's the same thing, right?
Sometimes. Of course, the graptolite literature goes back, 100 and something years, and sometimes the illustrations aren't that good. Especially, it has to be said, in Chinese literature when China was incredibly poor and didn't have much money. That they were doing their best with what they had, but the quality of the photographs are such that sometimes it's very difficult to tell whether yours is the same thing or not. But fortunately, we were in Nanjing, which has an amazing paleontology collection, including lots of graptolites. So what I could do is actually borrow what's called the type specimen, the holotype, from the museum, get photographs, and I can put good photographs of these holotypes in the paper.
Yeah, so
yeah.
The end result of all this is that if I have a new sponge, I might only have a couple of specimens of it, and I can spend a couple of days staring at it down the microscope, working out this little structure and how it all fits together, and then I just write it up, and I give it a name, and I draw a picture of it, and do all the illustrations, of course. And for Lucy, it can take weeks of tracking down random papers, going to museums, getting specimens out, photographing them, taking lots of measurements, and so on.
And for graptolites, especially at this age, because so many of the things look a bit similar at first glance, a lot of the species distinction is done by things like how wide the graptolite is, how many thecae there are in 10 millimetres. And to do that, you need several specimens ideally to get an idea of the range of variation. So I can't just take one specimen, unless that's all I've got, and just go, oh, yep, new species, whoop, done. I have to, first of all, decide which of all these graptolites I'm looking at form a species group, what I think is a species group, then go back to the literature to see how it fits in with what everyone else has defined. And since, taxonomy, there was a certain amount of opinion involved. Except for me, obviously, I'm thoroughly objective. And Joe, he's thoroughly objective too. But everyone else has a lot of opinion. And sometimes people define species slightly differently. And so you've got to actually make sense of all the existing literature, and it does take a while. Especially if it's in Chinese.
Yeah. She's actually got quite good at translation them.
Well, it's not so bad if you're just looking at graptolite descriptions, because the technical terms, there's only so many of them you need to know.
You go to the names, which are still in Latin, and the numbers, which are the measurements.
It does. It helps. And the pictures.
Right, well, yeah, I think that probably sums it up, doesn't it?
I think it probably does.
That's probably that's another 12 minutes on graptolites. You lucky people. Sorry. Won't happen again.
Oh, yes, it will.
Okay. Right, that'll do for now then, won't it?
Yep.
Alright, so those are interesting too. But it's obviously the sponges that are the star of the show, because these things are just unique. This whole fauna is really quite extraordinary. But I did also say at the beginning that this is telling us a lot about ancient marine deep ocean ecosystems as well. How can I justify that? How can I justify even calling this a Lagerstatten? Because graptolites are normal fossils and sponges have skeletons. Well, that comes down to how the sponges are actually preserved, because that's something else that is unique about Anji. In all the other sites across South China, the sponges are preserved mostly as the skeletons. The spicules are pyritized and replaced by iron oxides, and we're not seeing much in the way of soft tissue detail at all. But in Anji, it's a little bit different. What we're seeing is actually not the skeleton at all, but almost entirely the soft tissue. They're preserved as a combination of carbon and silica. They have spaces where the spicules were. There are moulds of the spicules, but what is actually fossilized is the soft tissue, including right down to the axial filaments inside the spicules, which are made of enzymes effectively, but they are preserved as carbonaceous, silicified carbonaceous remains. And we actually had a whole paper with a student at the time, Wu Suijin, who was putting all this together and working out how that fossilization happened. And it turns out to be a really interesting story. The preservation of the sponges was effectively very rapid pyritization. Pyrite crystals grew throughout all the soft tissues very, very quickly, and then redissolved again. So they're not oxidized, they're just gone. But when that happens, you end up with sulfuric acid being released. And that changes the chemical environment in such a way that silica, quartz, tends to crystallize. And luckily, there was loads of quartz dissolved in the water, dissolved silica ions, because the pyrite had not only grown in the soft tissue, it had replaced the spicules. So all the spicules and the silica of the sponges was forced out into solution into the surrounding water. Then the environmental conditions changed. You had this acidity coming in with sulfuric acid, which led to recrystallization, and it all went back straight into the sponges, combining with the carbon, producing this silicon-carbon organic material compound. And that is what has actually been fossilized. It's a combination of silica and the original carbonaceous material of the organic material of the sponges. That is not a normal process. And when you see that happening, you start thinking, well, why would that not happen to anything else that was living here? If there were worms burrowing through this sediment, we should see it. If there were arthropods here, we should see them. In fact, there was that one arthropod. We have this one sea scorpion that probably got swept out into this environment, didn't live here, and that was preserved in exactly the same way. It's a bit weathered, but it's absolutely the same type of preservation as we see in the sponges. We don't see any worms. We don't see any evidence of burrowing. It looks as though this was the genuine ecosystem. It was completely dominated by sponges, and we're not seeing other things there, but we would expect to see them if they had been there because of this preservational style. So that is actually telling us that we have this, in the late Ordovician, a deep water community which was absolutely dominated by sponges. As far as we can make out, there was not much else there. Maybe the odd radiodont lurking in the abyss, hence those few fragments that we have which might be from them. Maybe even the odd eurypterid, but probably that was living elsewhere normally. But we haven't got a fully developed ecosystem living in the deep oceans as we see today. And this type of ecosystem, the very deep abyssal plain type fauna, we generally have an extremely poor fossil record of. So having a window into this community at this time is actually really interesting. Of course it's limited. We don't know how representative it was of the rest of the world, but it's still a really interesting assemblage that we have here, which tells us things that we otherwise wouldn't know. There is a lot more to say about the Anji Biota in future videos, so I want to talk about in particular the implications of these extremely early modern-looking hexactinellids, and I really mean modern, and what that means for molecular clocks in this group, and how we can explain that not apparently working according to what our expectations were. I want to talk about some of the other sponges that have turned up, the wider ecology to some extent. So there will be more videos about the Anji Biota, but this was an introduction to it, to why it's important, what it can tell us, and why it genuinely does count as a Konservat-Lagerstatten, one of these sites of exceptional preservation, because it has tended to be overlooked by a lot of people, largely because it's dominated by sponges and graptolites, and that's a fairly niche audience for a lot of people. So there's a lot more to say about it, but it should be treated, like all of these deposits, as telling us about a particular community, not about the rest of the world, but it is telling us a lot more than you might think. It's not just a normal shelly fauna. This is a Lagerstatten, which is informing us about things we would otherwise not know. As a final note, at almost exactly the same age, as far as we can make out, there is another Konservat-Lagerstatten which is much better known. This is the Soom Shale, South Africa. And it was a totally different environment. We're looking there at a weird glacial lagoon habitat from near the South Pole. And there you have an utterly different fauna, but it's more or less the same time. So a direct comparison between those would be quite fun in future as well. But I'll leave it there for now. It's already quite a long video, and that's probably quite enough about sponges for today. It's never enough about sponges. What am I talking about? Right, on to more sponges.